Ladies and gentlemen and ladies, cats and dogs, children of all ages, and everything in between, welcome, welcome, welcome. You know who it is. It is the kid, Unc Just, the pizza freak, resonating at that pizza frequency. I love talking about pizza. I love making pizza, but mostly I love sharing pizza with people that I give a shit about. I mean, literally breaking bread with people that I care about. And I have a sneaking suspicion that you too, my friends, also resonate at said pizza frequency, or why else would you be here? And where is here? Dun 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 dun. Here we are. Slow Rise presents Science of Sourdough. This is part 6. Science of Sourdough, guys. This is a deep dive. We're deconstructing the genome that is the sourdough. This is going straight to my dome, the sourdough, right? It's an enigma wrapped in a riddle, wrapped inside one of those magic eye paintings from high school, 1994. Don't make a big deal out of it. Check out that hairline. That's what we're doing. We're trying to figure it out. We're trying to figure out different types of sourdough, what the taste, what the flavor, what the profile's on. Noel might bring out his flavor wheel. You know the deal. We've been doing this bi-monthly. In between office hours, we are attempting with Leaven Foods down in San Diego to basically sequence sourdough. And we're really excited. We have Speak Cheezy tonight and Bub and Grandmas that were so generous to donate some of their sourdough to the gentlemen in the lab, Cam and Jimmy. And we've been trying to figure out what the deal is with their sourdough, maybe where it comes from, what's the species, what's the genus, what's the bacteria, how that goes, and then maybe what those flavors, what their technique is, what their method is to coax all it out. I'm terribly excited. But without any further to do, I'm gonna bring him on, the man, the myth, the legend himself. The reason 'tis the season. I hope everybody's doing okay with the fall forward or backwards in springtime, spring backwards. I don't know. You know the deal. I feel bad for the animals. I'm gonna bring him on. Here he is, ladies and gentlemen, Mr. Noel Slow Rise himself, Brohner. Bluebell Noel.
Hello, Justin.
Oh, hey, bud. What's going down?
I got issues with your bell. I can see it, but I can't hear it.
Oh, so what are you saying? What are you saying? Just because you don't hear it, the bluebell doesn't exist?
If a tree falls in the forest,
I don't know. If a pepperoni curls and no one's there to drink the little fatty juices that come, I mean, like what is it? The
you're
eternal
fast today.
question.
You're fast. I thought I had you.
I'm fired, man. Fired. Because I missed you last week. We didn't have a class because you were down doing some dirty dancing down Sandeezy with my boys. We'll talk about that later. But it was also Halloween, and now we find ourselves falling backwards into springing forwards. And it's almost Thanksgiving. I can't believe this year is over.
I don't know what you just said, but I love the way that you said it.
There you go. What are we doing tonight? What's the deal?
Well, first off, do you have any pictures of yourself from Halloween?
I probably do. I'll put them up behind.
Let's
What-
let's let's
Yeah,
put those in the chat, please. I'm not
yeah,
sure they're safe for work, but
yeah.
safe
Oh,
for the chat, you think?
so yes, of course, of course. I don't know if I can. Yeah, let's see. I'll do that. I'll send it out to everybody in
Homework,
the chat at
homework,
some point.
homework.
Yeah. Thanks, guys.
And then
Um,
Cameron is asking about the link to the sourdough survey. Do you have that from last week, or you want me to find it for you?
yes, yes, I will put that up in just a moment. Yes,
Perfect,
I have. I
perfect.
believe I have it. I am putting in. Hopefully, everybody, guys, because we are doing. This is mostly a Q&A involving our participants and the our lab side of everything, the scientists. We're not going to be promoting people to panelists this time. It's mostly, we are going to be doing a little bit of a slideshow. So if you're watching, turn your phone horizontal. And I encourage everybody, if you do have questions, hopefully we'll have a chance to get to some of those at the end. Use the Q&A. Use the Q&A. I know in the past when we promoted attendees to panelists, we've just gone in the chat, but I think it works best for the Q&A. And I am typing, I believe right now, the form. Hopefully that's the form that everybody has access to. The Leaven Foods Sourdough survey. If you are curious about sequencing your sourdough. No?
We'll explain all that soon. Thank you, everybody, for showing up. We got a nice long list of people. It looks like almost everybody's here, which is pretty rare. It must mean that we got the goods this week, right, Justin?
I'm very excited. I'm very excited.
I'm very excited too. So thanks to everybody for showing up. Thanks in advance to Andy Cadine and Jason Winters. It's a crazy world out there, as we all know, and there's lots of things you guys could be doing. So thanks for spending just an hour with us on a Monday. As always, we're very excited to show you what we've been working on. As Justin mentioned, we didn't do office hours last week because I was down in San Diego with Jim and Cameron, having a good old time making sourdough bread and pizza with microbes. So I'm happy to announce that we did it. We've done it before, but we really did it last Monday and Tuesday. So good job, guys, in the lab. Those guys are incredible. So let's see. What are we going to do today, Justin? What are we going to do today?
I think we're going to bring Jimmy on. You guys are going to do a super quick, or Cam and Jimmy on, maybe do a super quick recap, and then let's dive in head first, shoulder first, feet first. So I'm going to go ahead and bluebell Cam and Jimmy if they want to come on, or just Cam.
I think Jimmy's hiding in the background. He's feeling shy today. That's all right.
Cameron, come on down, my brother. There he is. How you doing, bud?
Doing good. How about you?
Dude, are you kidding me? Look at me. I'm doing fantastic. Look at all these pepperonis I got to lick. Right? We are the music makers. We are the dreamers and drains. Pepperonis, it's like the schnozberry thing. Anyway, it's a Willy Wonka reference. All right, I'm going to get off. I'm going to let you guys do your thing. With love, everybody. Let's talk it up.
Thank you, Justin. See you soon, I hope. Cameron, how are you?
I'm doing good. How about you?
I'm doing good too. Thank you. Been a busy Monday, but I'm not mad at it. Lots going on in the world, huh?
Yeah. Yep.
Good.
Busy's good.
All right. So I've got my PowerPoint printed out, old school style, just so I can keep everything straight here. So it looks like there's lots to tell. So for people who don't know, I mean, how did you even manage to get all these beautiful sourdough starters into your lab? Tell us a little story.
Well, first of all, we emailed a bunch of people. You emailed people, and then I emailed them. And we were lucky enough that some awesome people were willing to give us their sourdough starters to explore and sequence. So James and I woke up at like 4 a.m. and drove to LA, and then spent the entire day driving around eating bagels and pizza and bread and collecting people's sourdoughs, and then got back here, probably in the late evening.
Now, when you say collecting people's sourdoughs, well, what did you actually collect?
We collected some of their starter.
How much? How much is some?
I think somewhere between 25 to 50 grams is what
Not
we collected.
very much.
Little conical tubes. I don't have one over here.
Not like little Dixie cups?
But no.
More scientific than a Dixie cup?
Yes. It's sterile, so there's no...
What were you saying? Dixie cups are dirty? I've been drinking out of them my whole life. They always seem clean to me.
There's probably some microbes. They haven't been gamma irradiated.
Probably.
So
they have not been gamma irradiated. Now you tell me.
We're just making sure we're not contaminating the good stuff.
Good. Good. So you picked up these sourdoughs. You put them in gamma radiated containers. Good to know.
Yes. Yep. And
they gave you also a little bit of
flour.
And what kind of flour did you pick up? Whatever they fed it with?
Yes. Yeah. Whatever they're feeding with. We also asked them about how they were feeding. We're going to get to that.
Good.
And yeah, should I share? Should I share?
You're welcome to share whenever you're ready to get going.
All right. Let me share. Let me make sure I'm sharing the right one.
You don't share the wrong one. It's not going to help. We only have an hour. We get off on the wrong path. There we go.
We're looking good. All right,
Pretty
let me
good.
put it into presentation mode, maybe.
That's definitely what we want, presentation mode. Because some people are looking on their phones, so they need all the. There we go. Nice job on the photos.
Yeah. Well, I didn't take them.
Oh,
I didn't cite my sources there, but
they look good.
So.
Is that a dog bowl outside of Speak Cheezy? Are they dog friendly over there?
Yeah, it looked. They're friendly to all folks. 4-legged and 2.
4-legged and 2. Good to know.
Yeah. Yeah, we're going to talk about these two today, who very kindly not only donated their starters but welcomed us in. We got to try some of some of the pizza, some of the bread, and it was wonderful.
Good.
But yeah, let's get into it.
Let's get into it.
So there's a few slides here from the last presentation we did 4 weeks ago.
4 weeks ago,
I think
yeah.
Justin's going to put the link into the chat, but we've already covered this a bunch, and I'd rather talk to them about how they're, feeding their starters, about their profiles, and, have a nice discussion about that rather than rehashing all of this. So if you're interested, there's bacteria and yeast in your starter. They're consuming the sugars and making the flavor. And we have a few videos about it. We have the microbial one that did a whole bunch. We've got more coming too.
So when you say we have a few videos, you mean that we've been making these YouTube videos for 5 or 6 weeks, and they're all on my YouTube,
yes.
right?
Yes. And Justin will probably put them in the chat, I imagine.
Very good. Very good. Well, one thing I can tell you is that a lot of people are here every week. I'm impressed. It's a good thing we're doing it on a Monday and not on a Saturday night, because I know some of these people are pretty busy.
Yeah. I appreciate everybody for being here.
Beautiful.
Okay. Should we talk about what we did in the lab?
Yes, please.
Okay, so we talked about it. We collected the flour, the starter, and once we were in the lab, we fed them according to the instructions that they gave us on how they feed their starter, which we're going to go through in detail and talk to them about, because I think that was really interesting last time we did this. And we took samples of the flour itself, so like what microbes were in the flour, the original starter, so the one we collected from them, and then we also sequenced and looked at the microbes in the starter that we passed here to make sure that we weren't altering anything once we got to the lab. Because we also, you can see here, while we sequenced it on the starter when it was fresh, so pretty much based off their instructions when they would use that starter to put it into their dough, inoculate their dough, as we would say in microbiology, but add it to their dough to make their bread or their pizza dough. We also did cell culturing, so we isolated their microbes and we counted the number of viable, which we talked about previously, but that's the number of microbes, both yeast and bacteria, that could make more of themselves. So they're alive. They're not, sometimes they explode, and then you don't want to count those ones. Questions? Did we cover this? Cover it okay?
I think we did.
Okay. I think we need to bring James on, because James did the feeding. So I'm gonna let James explain what we did, and then we can talk to each person about, did we do it right? Did we follow the instructions okay?
Good.
That sound good?
Yeah, that sounds great.
All right. How you doing, Noel?
James, good to see you.
Likewise. Just glad to be here and talk about these starters. So it was pretty cool to get them into the lab and see just how people out there are doing things differently,
Gotcha. So you got these instructions from the actual bakers themselves, right?
Yeah, from our survey.
Good.
So at least as well as I can interpret it, which I think we'll see how I did.
Good.
See how I did. Yeah.
So tell us about what we're looking at.
Yeah. So this was for Andy's starter here at Bub and Grandma's. And so, as Cameron said, we got the starter into the lab at, 7 o'clock that day, but we might have collected it at 10 a.m. So it had been sitting in the car for a bit. So we really wanted to make sure that, we refreshed it and then we fed it the next day kind of fresh, the way that they would handle it in their bakery, So with this starter, we refreshed it in the lab by taking 10% of the original starter that we picked up at the bakery, 70% hydration, and then room temperature water, and then incubated it overnight at 60 degrees in the fridge. And then the following morning, we repeated the same process. So I really like this feeding schedule because it's just simple. It's just very easy. And I think the starter was super active, and it seemed to be very effective, and it was cool that it was, we've seen some starters at room temperature, and we've seen some get stored colder, and this was kind of like right in the middle. So it seemed to work really, really well.
So let me just ask one question. So you say you see some at room temperature and some refrigerated. This one we refrigerated in a refrigerator that held it at 60 degrees Fahrenheit?
Exactly. And so some of the starters that we saw from the last time we did this, Giants and Clark Street, they were keeping theirs more at room temperature. This was not necessarily cold, not 40 or lower, but it was 60 degrees, so not exactly room temperature, not exactly cold. So I think it was really active the next morning, and then we fed it again and waited towards the end of the day to sample it again, 12 hours from that point, and really active again. So
good. So Andy's doing relatively low percentages of starter compared with the flour weight, which would be 100%, which is implied but not written, right?
Yes. So this was the 70% hydration, so 70% of the flour weight.
Good. All right, looks good.
Yep. So does Andy want to come on and maybe talk about it, or are we gonna hold on to do that?
That's a good question. How about this? Why don't you do this? Why don't you next present the starter from Speak Cheezy, and then we'll invite both these guys on and we'll talk to them both. How does that sound?
All
we'll
right,
bring Cameron back on as well.
That sounds good.
Good.
All right, next slide, Ken. All right, so the next one was from Jason at Speak Cheezy. This was also really cool. So we refreshed the starter when we returned to the lab, same at 7 o'clock that evening. 10% of the original starter at 100% hydration with warm water, and then incubated at 45 degrees overnight. And now I know Jason, from reading the survey, he doesn't necessarily have a seed percentage. 10% was an approximation. He's more on like just a discard, which I think is fairly common. But for science, we kind of made an approximation. So we guessed 10%. The next morning, 9 a.m., same thing. We fed again, 100% hydration with warm water, 115 degrees, 10% of the starter again, and then we incubated at 45 until it became active again. So we were really trying to take these samples when the starter looked like it was going to be active, like when they would use it, and so I think this is another, this was cool too. This was another unique way of handling these starters that we hadn't seen so far. High hydration, which was similar to some of the ones in the past, but the feed with warm water. But the main unique thing was incubating at 45.
Yeah.
I think Jason actually incubates it a little bit cooler, maybe somewhere between 39 and 41. But again, this was, super active starter. It took a little bit longer to develop, because we fed with that warm water, but then it goes into 45-degree refrigerator. But yeah, really cool and just a different way of doing things. So it was really cool to see that.
Got it.
Yep.
Beautiful. All right. Thank you for the explanation.
Mm-hmm.
A lot there, but I think we got it.
Good. Want to move on?
Yeah, why don't we bring Cameron back on? And Justin, if you want to ring that blue bell,
you know that I do. You know that I do. All right, so I'm going to ring the blue bell. I'm going to bring James and Andy on. So I'm sorry, Jason and Andy on. So guys, once I ring the blue bell, go ahead and turn on your videos and turn on your microphones. Here it comes. Thank you. Blue bell of pizza-ness. Whoa. Beautiful. Hello, Andy. Hello, Jason. Jason, can you make sure that your microphone is on, brother?
Hello.
All right, perfect. All right, well, I'm going to bounce off again and let this boat get on the show.
Beautiful. Cameron, you back?
I'm back.
All right. So where shall we begin?
I mean, how did we do, guys? Did we match what you're doing? Where did we miss?
Almost.
Almost.
Andy, let's start with you.
Can you go back a slide so I can riff off of that?
Yes, please.
It's very close. I think the unique thing for us is the 60-degree overnight. And you know, we're sort of there's a unique issue that's kind of created by the health department that we are sort of skirting with that. We have the unique privilege of having 2 gigantic walk-ins in our wholesale bakery, and one of them we keep at 40, which is where all of our you know, cold proofing sourdough and a variety of other breads hang overnight. But we also have another one that we keep at 60 degrees, specifically for the purpose of you know, retarding our 10% seed overnight. So that part is correct. 70% hydration. We like a stiffer sourdough, which helps us to a have a more workable product. It's less complicated than a liquid sourdough to get into mixer bowls, but also it produces less acid. So you get a little bit less of a sour sourdough in your final product with the stiffer starter. The part that's a little bit different is the feed. And I think this is just maybe that you guys were working towards uniformity of the feed at 9 a.m. and then you can kind of determine when things are active. For us, it typically starts at 4. Right? We do the leaven mix first, so that 10% seed gets mixed with the remainder of the flour at 70% hydration. And then while we're mixing all of the autolyses for all of the other doughs that we have to go through, that's when the starter develops and gets super active, and we mix it with water that will take it up to about 80 degrees so that it can optimize and be super crazy busy and hungry when it gets portioned and mixed in with all of the sourdough mixes that we're doing. But yeah, so the 60 degree is the super unique part that we kind of you know, we were looking at wine fridges and a variety of other things that we could do to try and keep it not necessarily in the colder realm, but also not in the room temp. The room temp realm is difficult just because you have no control over what the temperature is going to be overnight. Yeah. So if we're talking with trying to keep things in that middle ground where it's developing, but also like retarding a little bit, 60 degrees kind of was this middle ground where we felt like we could keep control over things.
Gotcha. So where we got it wrong was the temperature of the first feed was 60, but your desired dough temperature of the second feed was actually 80, and so you use warm enough water based on the room temp and the flour temp to get you to 80. Correct?
Same. Those temperatures always vary depending on, you know, if we're talking about fall or summer. You know, the room temp is wildly different. So the bakers will always try to compensate to bring things closer to 80 just so you have a steady spot to move to. But yeah.
Good. All right. Thank you for that. That's super important. I noticed that a lot of people just feed, for instance, their starter at, for some reason, 115 seems to be a go-to temp. But as you said, depending on what season it is, the room temperature goes up or down. And so that means you have to vary your water temperature as well, right?
Yeah, you just stay flexible. Yeah.
Stay flexible. I love it. Beautiful. Thank you for clearing that up. Let's see here. What about you, Jason? How'd we do on yours?
Pretty good. We are more equal part starter, water, and flour. More of a, I guess, the pizza maker's method. But I really do appreciate everything that Andy said. I learned a lot from that, to be honest with you. Maybe doing a 10% or something might give us more play at room temperature, but I think it originally kind of started that way when we were catering, because our events would be on the weekend. We'd be in the kitchen at, days before preparing the dough to do our 5-day ferment. And it was easier for us to, refrigerate our bulks and then ball the next day and make the days short and spread it out so that they are ready by the weekend. So that's kind of how we developed our method. But we're more equal parts. We take the starter out in the morning from the refrigerator. We create the leaven from that. And then we the way that we create the leaven from that is we feed it the hottest water out of the tap. We then temper the cold starter that's like 39, 40 degrees until it feels like our body temperature. So usually we'll get up to like around 85, 84, 85. Then we'll discard, and then we'll feed it flour, and then we'll use that for our leaven. And then we'll also put a portion of that. We'll let it sit out for about an hour, hour and a half, and then it'll go back into the refrigerator. And our starter is very consistently or our leaven, it's usually a 3.5, 4 hour window consistently throughout the year that way. Whether it's summer or winter, we're able to kind of manage that part of the process very easily from cold. We do a similar, I guess, more of a bread making technique with the autolays, where we'll feed the starter or multiple starters for however many batches of dough that we're making that day. Or we'll mix our dough first and then feed our starters. And then we'll pull our dough from the mixer, put it into Lexans, keep it in a cool environment while our starters are growing. And we like our dough to come out of the mixer at around 67 to 73 degrees, because we're going to be balling it the next day.
So if it comes out too warm, it'll rise too much in bulk. Yeah.
It will. Yeah, we're definitely taming it. So I think we're going for the long and slow across the board, technique with the pizza, right? So I think as a bread maker, like when we do our pan pizzas, our squares, it's more of a bread maker technique. Like we're feeding it 85-degree water. It's a wetter dough. It sits out all day. We portion it at night. We put it in the fridge, and then we bake our pan pizzas the next morning while they're after letting them sit out for about 3 hours. And that develops a really great flavor as well. But I think there's more whole grains in our pan pizzas than our regular pizza.
So you have, how many styles of pizza do you do at the shop?
Just two.
Just two. So you do
there used to be more, but just two for now with the
just two. And how do you call those styles?
I just call our 14-inch round California style right now.
Yep.
I think obviously there's a unique style to California that a lot of us are really happy to be a part of right now. And you've played a huge part of that as well. So we're all happy to be here, I'm sure. Andy, real quick, I had a 55-degree refrigerator that broke down.
Yeah.
We put the shave ice machine there and got a low boy freezer instead. But I used to love that thing because we would keep our starter in there. And we would also put dough balls in there if we wanted them to proof a little bit more or faster. We would manipulate them in there. And the 55-degree was amazing to manipulate. So I think pizza makers should have two fridges, a 40-degree and like a 55 or 60-degree as well. Right.
It makes sense. It's a super useful tool that, the health department may not like that much, but everybody else does.
And that's how you raise your yeast count.
Yeah. You can get, it's not going to hold it all in one place, and it's not going to let it go gangbusters.
Mm-hmm.
Excellent. I'm just curious, Andy. When I first met you, you were in a very small bakery in East LA, and I believe you only had one very packed out walk-in fridge. That one was on the cold side, right?
It was on the completely all over the place side. Yeah, we were working with really under compromised circumstances in the beginning. Not to mention working when it was like literally me in my house, which is how it all kicked off.
When did you make the change from to two walk-ins at two different temperatures?
Probably like right around just pre-COVID, I think, is when we moved into our current bakery, the wholesale bakery on Riverside in 2018. So somewhere between 2018, I think it was probably around the end of 2019 that we made that move, because we ran out of space in the smaller of our two kind of walk-in spaces, turned one of the larger spaces that used to be production into a walk-in.
Right.
And then now we have the two to kind of work with. It's also when it's 110 degrees in a bakery in the summer, that's everybody takes their breaks in the 60, which is amazing.
So you say you store your starter in there. Do you also store your dough that's leavened with starter in there?
No,
No.
all the doughs. Well, when we mix the doughs during the day, they're either going to bulk retard overnight in the 40, or they're going to be shaped and then sit in racks to be baked the next morning in the 40 overnight. So all the dough goes into the 40 overnight that's produced. It's literally just for, maybe at this point a dozen 8-quart Cambros that are sitting on a cart. Plus, we keep all of our seeds in there, our raw seeds. We keep other kind of physical storage, like all of our pizza boxes, of which we have a ton of, and other things like that.
Because you're doing dough for Nancy Silverton as well, right?
Yeah, we do the pizza dough for Pizzeria Mozza.
Beautiful.
That's badass.
That is badass.
Andy, I'm gonna follow you right now. I'm looking for you on Instagram.
Oh, sweet. Thanks, man.
Follow all of his accounts. He's got a few.
Okay.
No, just one. I mean, I have my account, but there's nothing.
Wait, let's call it out right now then. Crap Catapult.
Yeah, that's me.
That's you. Crap Catapult. All right.
Yeah. Misspelled on purpose.
Like I said, let's follow all of his accounts, but you don't have a separate account for your what do you call it? Is it a sandwich shop? Is it a deli?
Restaurant?
Is it a restaurant?
Yeah, just restaurant. I mean,
restaurant.
It's got labels are tough, I guess. I don't know.
Labels are so tough.
Doesn't need one.
So, guys, we have a lot of stuff that we want to get into, but let me just ask you one thing before I pass it back over to Cameron. One of the things that we wanted to do tonight was actually spend a little more time on you guys and your starters, different from the way that we did it with Clark Street and Giant Isaac. And one of the things I'd just like to ask you both, Andy, I'll start with you if you don't mind, is how has your, let's say, recipe and process just for your starter changed since you started, maybe even working at home or at your first very small location?
It's pretty weird. I started the format of making sourdough, mixing your leaven, letting it get excited, mixing it in with the remainder of the dough, letting that do its thing, and then shaping, proofing, baking is exactly the same. Whether I was making, 5 loaves in my home oven and, exploding my oven light bulb, or now we're making thousands of loaves a day by a bunch of people that aren't me. The process is very, very much similar. The difference is, actually at this point, we're kind of at this sort of pivot point where we can't fit any more bread into that 5 a.m. to 12:30 p.m. kind of standard bakery schedule. We have people coming in at 2:30 in the morning to bake off as much bread as we possibly can, but we can't add a single other baguette because it just doesn't fit into that approach to making bread anymore. So it's evolved, but it's still basically in the same format that it's always been. The bread making process is the same. But a bunch more people, a bunch more bread being made in that same format, and we're about to kind of blow that all up and kind of create these overlapping shifts that are going to allow us to produce more. And the only way that we can do that is by, layering that same process on top of each other. So you have sort of schedules that overlap in the middle that allow us to produce a great diversity of different kinds of breads within a fixed amount of time. But yeah, it's the same process, but it's stretched and widened.
Stretched and widened. And just out of curiosity, did you always refrigerate your sourdough at 45 degrees, meaning your dough that was leavened with leaven, or did you ever do a
shaped bread?
Shaped bread or bulk bread? Did you ever do it?
I mean, I've tried it all, especially at the beginning, especially when it was just me or a couple people working for Bubs. It's changed. We've tried all those things. The overnight room temp is extremely difficult to regulate. And that's the part for us, is how many variables can we isolate and make uniform. So over the years of developing our process, the more ways that we can make our process uniform and the temperatures of the doughs uniform throughout the process, the more you're going to see a result that is the same as the day before. And that's our goal, especially as like a business, which I never thought I was going to be. the only way to do these things is to try and create consistency from day to day, so that your customers, which are restaurants, can get the same size product with the same lift and the same flavor and the same everything inside. And the only way to do that is to isolate these variables and try and get them to perform in the way that you want them to. So that's the part that's developed over time.
Awesome. That's a great description. Thank you.
Yeah.
Cameron, you still there?
I'm here.
Good.
I'm learning.
Good morning. Why don't we check in with Jason at Speak Cheezy, and I'll ask him the same question as well, as to whether his process and recipe have changed, and then we'll go on with the rest of the presentation. What do you say?
Sounds good to me.
Good. Jason, you around?
Yeah, I'm here.
So same question to you. How has not your process and recipe changed with regard to sourdough since you started making either at home or at your pizzeria?
Yeah, I can kind of tell you the exact time it changed. It was very quickly into towing around a wood burning oven outside in the elements with no refrigeration. We did, I think his name was John Voltari recipe from which was a hybrid dough, they called it back then, where it was a mixture of starter and man-made yeast. And we started that in the very beginning. And we were in the center of Pershing Square, and it was really hot, and the sun was just beaming down from all the buildings around us. And I just looked at the guys and I'm like, fuck the yeast, no more yeast, just use the starter.
Yeah.
And the second that we started using the starter, the event was for Headley and Bennett, and we were doing a fundraiser for Wiser Farms with the Tehachapi Grain Movement. And, we always used a sourdough starter with small yeast. And I think a lot of people still do that. And we're actually getting back to that, right? Like small amounts. But it was kind of at that moment where it was just fuck the starter and, oh, hey, here's these local grains that are available to us. And that was through the fundraiser at Headley and Bennett that we did. And it was great. I mean, I could take the dough out in the summer with the keep it cool bags with no ice blankets. And it was very easy to manipulate. And so from then on, because we were stretching fresh at events back then, it was just a lot easier for us to control.
Yeah, it's beautiful. Some people complain that sourdough is a little sluggish. It takes too long. They're waiting. It doesn't rise. But if you're outside in the elements and you've got hot summer days and direct sunlight, it's actually kind of a godsend, wouldn't you say?
It is, yeah.
Beautiful. All right. Well, thanks for that description. That's super helpful. Cameron, let's have a look at our presentation. We've got not a ton of time, but I know that you're pretty quick, so I'm not worried.
Yeah. Okay. So the first thing we're looking at is the bacteria. So you got bacteria and yeast in your sourdough. Both of you have pretty much the same species between the two, which is the Fructilactobacillus Sanfranciscensis. But labels are hard, right? We just covered that. Labels are hard in microbiology too. You can see, you've got these nice rods here. There's your microbes at 2,500 times magnification. So they're very, very small. And this bacteria, at least the species name, right? So we've talked about this before. There's species, which is a huge, broad classification, like calling something, in the Canis family, a wolf, or in the Canis species, a wolf and a dog, right? So you have a huge group of things that could fall into a single species that are very different. Like your wolf and your Labrador are two very different things. One of them you want in your house and one you don't. And just like that in the microbial world, the same species might not be the same thing in terms of how it works in your dough. But this species is pretty common. So 1 in 5 sourdough starters most likely will have this Fructilactobacillus Sanfranciscensis in it. And when it's there, it completely dominates. So it does a really good job probably of doing a fermentation in the dough. It's never been isolated from anywhere other than sourdough. They've sequenced it in bugs and in flour, but they've never been able to isolate it. And like I was saying, the strains can have very different impacts, right? So for instance, here we're looking at different strains on Petri dishes, and some you'll notice are like really white looking. So they can produce these polysaccharides in their cell, which can even change the texture of your dough. It can change, the way your dough works. So that's just to say, even though they have the same species of bacteria, which is interesting, the strains could play a big role, which we just don't have the capability to do strain level classification.
Not yet.
Not yet. I'm going to skip this one. We've talked about this one multiple times. We have a whole write-up about it. But it's just to say that your microbe here falls into the obligate heterofermentive category. So it's So it's producing either, depending on whether it's consuming a pentose sugar or a hexose sugar, it's going to produce lactic acid and acetic acid and CO2. It could also produce lactic acid and ethanol, but it's very, very most likely contributing to the rise of your dough as well. So it's not just your yeast in there that's contributing to the rise, it's your bacteria too, is making gas for both of you. You have two different bacteria. Sorry, the graphs are both orange. It's kind of misleading, but you can see here with the labels. Mm-hmm. You have in Bubs, you've got this Kazachstania humilis, which used to be called Candida humilis. Candida just means we don't know where it belongs. We can't really fit it into the phylogenetic tree of life, so we're just going to throw it somewhere. And then a Saccharomyces Cerevisiae. Saccharomyces Cerevisiae, also the name of the species in baker's yeast, but again, strain matters here. I would hazard to guess this will behave very differently if we were to isolate it and grow it up than your baker's yeast would. When you find this bacteria, so right, this pink here was all that Fructilactobacillus Sanfranciscensis, and now I've added the yeasts under here, and each one of these bars, right, represents a different sourdoughs. There's 500 of them plus here. The Kazachstania is in this light blue. You can see it kind of showing up here in a high percentage. And the Saccharomyces Cerevisiae is here in this kind of goldish tan color. And that you see shows up everywhere. Saccharomyces Cerevisiae found a lot in sourdoughs. Again, strain likely different than the baker's yeast strain. But when there is this bacteria, you see this Candida or this Kazachstania humilis and other Kazakhstania show up a lot. And there's a reason for that. We covered this a little bit in the last video as well. But the Kazakhstania humilis that's in Bubs is maltose negative, most likely, meaning it can't eat the primary source of food in the flour. It's completely reliant on the bacteria to break down that maltose, and the bacteria feeds it glucose. And so this is a really strong relationship between a yeast and a bacteria found in nature. It's really interesting. Microbiologists very interested in the relationship for lots of reasons, but the yeast likely feeds it back amino acids and vitamins and other things to keep the bacteria happy and continue feeding the yeast. We don't need to get in all of this, but this is just to say that the Saccharomyces strains are not all the same. So for instance, here in this plot, each one of these dots is a different strain of Saccharomyces Cerevisiae. We've got commercial strains like the ones that you find in baker's yeast. We've got sourdough strains that were isolated from different sourdoughs. We've got wine strains here, beer strains. We've got other fermentations here. So like things that are found in completely different types of food fermentations. You can see just the CO2. So this is the level of CO2 produced in a fermentation. Just the CO2, especially in the wine and beer, is way lower, which you'd expect, right? Wine and beer, you're trying to make alcohol. So you're not necessarily trying to select organisms that make a ton of gas. And this has probably happened over a very long period of time of people using these fermentations and not even realizing they were directing the evolution of these strains, right? Same thing for the difference between the sourdough and the commercial strains of Saccharomyces Cerevisiae. They have differences. So not only in the amount that you find in dough, but in a lot of different characteristics, including the ability here is just showing maltose growth, the primary sugar in dough. Strains pulled from sourdough work much better in maltose. They can grow on maltose really, really well compared to strains pulled from all other environments, which makes sense. They're adapted to grow in dough. You can find these strains in lots of different places. So like I said, the bacteria that we found, this Fructilactobacillus Sanfranciscensis, it's been sequenced. So they've taken a sample from grain beetles and plants and things like that, and they see it in really low abundance, but they've never been able to isolate it. It's primarily found in really high abundance in the sourdough. So at some point when you're enriching for the microbes, right, when you're making your sourdough, when you start it, for some reason, this bacteria really does well in that environment and shows up a lot. And then both the Kazachstania humilis and the Saccharomyces Cerevisiae are found on a lot of different plants and in a lot of different fermentations. Kazachstania is found in other fermented foods like kefir, wine, kimchi, even in beer. And Saccharomyces Cerevisiae is used in a lot of different foods, and you've probably heard it in other sources. It's used in beer. Other strains are used in beer a lot. But something really interesting about these is that they most likely rely on insects for their dispersal. So they insects lands on a plant and it carries these yeasts from plant to plant, and the yeast gets dispersed all over the place by hitching rides with bugs. So I think this is a really good place to have some discussion. And I think maybe if we were to do Andy's bloom there right at the right before he's going to add it to his dough while he's doing his autolyse, this might change. But what we're looking at here, so on the x-axis of each of these plots, we have the number of microbes. So this is the number of viable cells per gram of sourdough starter. So the number of cells that could do something when you add them to the dough, right? We're not counting any dead cells or dormant cells, just the ones that could perform the chemistries that make your dough taste good and give it the rise. And this is measured in log scale. So there's a ton of microbes here, not as much as you'd find in your commercial yeast per gram, but we're starting at 10 to the 5 here. So 10 to the 6, you get 1 million, and then we're going up to 1 billion every 3 here. So the number scale is very, you have a lot of cells in there. Both of these are pretty close to the average. I imagine if we were to bloom Andy's starter, the amount of bacteria to yeast would get closer to this average. I bet it encourages the yeast growth right before he's inoculating. Without doing that, we had a higher amount of bacteria to yeast than the overall average, but it's not well outside things that we've seen before. That what's commonly observed is somewhere between 100 times to 1,000 times the number of bacterial cells to yeast cells in a sourdough starter. Jason, you're right on the money in terms of what people have commonly observed in terms of the number of viable cells that is in a sourdough starter before it's used to make bread. That's somewhere around 100 to 200 times the number of bacteria to yeast, and you're adding somewhere over 10 million yeasts and 10 billion bacteria. So you're adding a lot of cells per gram of your starter that you got in there. I think this is also really interesting to contrast to what we saw with Giant and Clark Street, who had these 2-step feeds that we talked about 4 weeks ago with really low bacteria to yeast ratios. In particular, Clark Street, they added salt to bring theirs down. And Giant did it by the feeding schedule, essentially feeding, right out of the fridge and getting it super active right before he made his bread. But yeah, I think it's pretty interesting to see the differences in the not only the number of cells, but yeah.
I think because I do kind of the colder, I'm able, even with my starter, right? My starter lives in the fridge 23 hours out of the day, and then it's fed warm water. So I think that's why the bacteria is more prevalent in there. And maybe it's the 30%. I don't know, maybe the 30%. Because it's colder, that's where the microbes thrive and the yeast stays dormant. But then you look at my yeast, and my yeast is also up there. So
yeah, you have a good active starter, I would say.
Okay.
All the starters were super active.
I think it also, just to say one thing, that I think it also maybe organically develops this way relative to the process, at least relative to bread, because of what you're looking for in the end goal.
Mm-hmm.
And not that there's any better or worse approach, just different. A lot of people are aiming for, in the bread world, a much more open crumb, a much larger holes developed from much larger production of gases.
Yes.
Same thing somewhat with pizza dough, where you want to have a crispy light crust that has nice open holes in the crust. But for us, for our bread, we like to keep the development to much smaller bubbles, and maybe that's why, without ever knowing that our levels were on the lower side, why we've landed on that is just because we prefer a tighter crumb inside our bread. Yeah,
a nice perspective. Yeah.
One thing I'm curious about, when you guys both started your starters, do you remember what kind of flour you used at the very beginning?
Like the very, very first time?
Very first time.
I do. Yeah. I mean, I think that the percentages have changed, but it was, I think it started at 70%. At that point, central milling, what's it called? What's the
ABC.
standard? Yeah, ABC Plus, I think, was what I started with. I don't even know if they make that anymore.
They do.
They do, yeah. And then some grist and toll hard red at 30%, so 30 to 70. Now we use Galahad for our bread flour, which we love, and 30% Camas hard red. So in grist and toll we use in smaller percentages in other breads, but at the volume we do, we had to switch to larger producers like Camas and KA. But that was what we kicked it off with.
Now, do you remember, Andy, when you first made your starter, did you get it from someone, or did you start it from scratch?
No, from scratch. And whenever people ask us to buy starter, which happens often, I don't. It's not like to be a dick. It's that I just don't. I'd rather people, use a recipe, and we have a recipe on our website that we built for designing their own leaven, because it just kind of feels more. It takes 5 days, 7 days, and it feels more your own than borrowing from someone and kicking it off. But at the same time, there is no invalid approach. There's just like, if someone gave you a sourdough that's they've had for years, like, why not just kick it off with that? Or a grape leaf or anything. Yeah.
And when you initially built your own starter from scratch, were you using the same flours that you were using to fill it with, to feed it with?
Yeah. It was central milling ABC Plus and Nan's grist and toll hard red.
And the hard red was like a type 65, 85. What was it?
Everything's whole grain.
Everything's whole grain.
All of the whole grain flours are whole grain. We don't use any sifted flours at the bakery. Just bread flour, but that's in a roller mill. But bread flour and whole grains. That's it.
Good. Got it. And what about you, Jason? Did you use a combination of flours when you originally built your first starter?
It was, I think, think I was using 00 in the very beginning because I was more focused on wood fire pizza, like Neapolitan style. But we would feed it ABC Craft Plus was our starter. And then we made 00 was the flour. And then the very beginning was a little rye.
A little bit of rye. And when you, not when you originally started feeding your starter, but when you actually built it from scratch, what flours did you use?
Oh,
same ones?
I got it from my mother.
Got it from your mother?
Yes, yes. My very first chef I'm still friends with. I was 15 years old, but I also gave it to him. And at one point, when we were in Commerce, I lost it and I was able to go and get it from him again. He still had it.
Got it.
Yeah, so she got it from like the friendship bread.
Got it.
She was my mom would bake bread and make soup. That was her thing.
That was her thing.
Mm-hmm.
So Cameron, I'm just curious from your vantage point. It sounds like Jason doesn't know what flours were used to create his starter from scratch because he got it from his mom. But is it safe to assume that it might also be like Andy's, where it's a combination of whole grain and maybe white wheat?
Yeah, maybe. I mean, it's really hard to say. There's just not enough research to be able to track back and figure out, you'd have to sequence every flour and every grain that ever existed and then backtrack it back to figure out exactly which strains.
I have another question. If we took some of Andy's starter and mixed it with mine, would we have his bacteria, mine, and ours, and would they thrive together?
That is an extremely insightful question that microbial ecologists are extremely interested in. And actually, I think on the 20th, we're having Angela on, who is a professor at Syracuse who's actually studying that exact thing. So I'm actually interested to find out on the 20th too, what she's found, because I don't know. Yeah, no one knows what will happen. But I mean, I think the beautiful thing about these starters, like Andy was saying too, is that they kind of become yours, and the history of them matters just as much, even though, it's nice to know what microbes are in there and what the viability is and things like that. It's like a important piece of both the history, and it's kind of, I view it also like a microbial pet, but that's just me. But it's like my dog is a boxer. There's lots of boxers in the world. They're all over the place, especially around my house. I've seen a bunch of them, but it's special to me because he's my boxer. Even though there's a bunch of species, but a bunch of other of his species roaming around the neighborhood.
Yeah.
And these viability counts, I think, too, also kind of match the practices in the way, like Andy was saying, in a really interesting way. All of these like ways of controlling the levels of the bacteria and the yeast match the way that the user wants to make their end product, which is also really cool. It's like you're adding, when you're using commercial yeast, right? You're like using percentages and it's all controlled. You're doing the same thing here with your yeast and the bacteria, but just without measuring it, just by feel, which is pretty incredible.
It is incredible. All right, then. How are we doing with our presentation there? We're almost there?
I think that was it. That was it.
Right.
Unless people have questions for me. And thank you guys for sharing your starters with us so we can look at them.
Thanks for, it's so cool. So it both answers a lot of questions and brings up a lot of new ones. So it was very exciting to see.
I was asking Cameron over the phone, can we measure our bulk? Can we find out the pH level or the bacteria? And then can we measure our dough balls? And can we measure the pH at a certain level? Like, is there a tool? And I think you said you might have done some research on that, Cameron?
Yeah, I mean, there's no true way to measure, I think it would be hard to measure the viability, at a location without, you could do it if you want to buy Petri dishes and, do serial dilutions and plate them on petri dishes. You know, maybe there's some undergrad over there, maybe at UCLA or one of the universities around there, who would be happy to be in a bakery instead of a microbiology lab doing that.
All right.
But I think, Andy, you introduced me to the breadwinner guy, who's and I think that tool might actually be, you know, pretty useful. We haven't tried it yet, but
I mean, I think he's also just at the beginning.
But
just for anybody who's interested, a friend of mine, Fred Bennington, is developing a product called Breadwinner that is a way to track the optimal time for using your leaven. It's basically you build a starter in a I think it's like a 16 ounce or it's built for a 16 ounce ball jar, like a glass jar. And it has an attachment that you screw onto the top that can kind of sense the development of the starter and then send you messages when your starter's at the optimal time to take. And that's just the beginning of the things that it could sense. Just seems like a really cool platform for that's tied into what you guys are working on, which could be really cool.
Yeah, because then eventually you could be adjusting the temperature. A computer system could be adjusting the temperature of your refrigerator.
Totally,
like a Traeger grill, pellet grill.
Yeah, I mean, we have a thing on our it's less about like fluctuations and more about if our cooling system craps out. But there's a thing called iMonit, that is a sensor system that you can put inside your walk-in cooler or a bunch of other kind of modules that they have to sense when there's any fluctuations in the temperature. And if it drops below a certain threshold, it sends you like a crazy alert that wakes you up in the middle of the night that says basically your power's out and your walk-in's not working and go bake your bread.
Well, I'm on it. I'm on it after that.
Yeah.
I mean, I think it's
go ahead. Sorry.
No, go ahead.
I was just gonna say the only reason why we have it is because it happened before where all our, the walk-in died, and when they come in at 4 a.m. to start baking, everything's pouring over the side of the rack and is garbage. So this is a way of protecting ourselves against those things that have happened multiple times in the past.
Every time my power goes out, my staff comes in. I'm like, don't worry, it's sourdough. It'll be on in like 6 hours. They're not used to it. It's the man-made yeasted days, right? So
just pour over the sides.
Yeah, beautiful. Cameron, I got a few questions from the crowd there. Can I spoon feed you a few?
Do my best.
Good. So Sirhan is asking, how can I maximize the milkier flavors and minimize the tang from my starter? Looking for more lactic than acetic.
Well, I think we got two guys here who could answer that better than I can. But from a scientific standpoint, I think hydration is probably one of the easier ways to do that. If you increase your hydration, you'll get a more lactic. That is also dependent upon the species of bacteria that are in there. But
I told him the exact opposite.
Well, hold on. It really depends on your starter, as we found. Andy, you're not wrong. There's not a right or a wrong. It depends on your starter. Different starters react differently to hydration. We found that out the hard way, didn't we, Cameron?
Yes. Yeah. We talked about that a few Zooms ago. Yeah.
Do you remember which Zoom that was? Because we don't have time to cover that. Sorry about that, Andy. Do you remember which Zoom that was?
I'm not sure, but I can find the link.
Find the link and send it to Andy, because I think it's really interesting, Andy. Really,
I'm curious.
Which one?
Yeah.
We were trying to remember which Zoom it came from where we were talking about this issue. Don't worry about it now. We don't have enough time to cover it. We'll figure it out. It's a really interesting topic that came up, and it's always beguiled me that some people would say by adding more water this would happen and less water that would happen, and the opposite would be true with other people. And these were all like professional bakers.
I think it also had something to do with temperature too, if I'm correct. That that one of them created more acid and one of them created less temperature or more temperature, created more lactic versus the
low and slow versus a quick rip.
Because we want it milky, we want it that, or some people want the lactic flavor, but some people, I mean, there's no. What I love about it is it means so many different things to so many different people that it's very taste driven. Some people want the more acidic, but some people are looking for the more milky lactic natures. But that's just, I mean, pizza is subjective, right?
Thank you, Justin. So I think, Cameron, I think you gave a good, quick answer to that. And unfortunately, we don't have time to ask the two pizza makers what they would say. But also, let me get to my second question, which actually kind of dovetails. Brian's asking if you want to increase the sourness of your final loaf of bread, what would you suggest? More levain in the final mix or something else?
Sorry, one more time. I was still thinking about the hydration. I was trying to
That's
remember
okay.
which bacteria.
That's okay.
My mind was in another place.
Brian was asking if you want to increase the sourness of your final loaf of bread, what would you suggest? More levain in the final mix or something else?
Oh man, I defer to the pros down there.
Yes.
Colder, slower.
Colder, slower. Got it.
You'll develop more. I think that's where you get more of the sour.
And I think, just to jump on that, I think that's what we talked about, that the yeast loves it warm, and that gives you the lactic, and the bacteria likes it cold, which gives you more sourness.
Yes. The yeast likes 55 degrees, and the bacteria can still thrive lower, but just they do it slower and longer to catch up to the yeast, I would say. So that's kind of what we do. We do really long and cold, and we tame the yeast. We want the yeast to be ready in 5 days, but we want the bacteria to be up there. And for me, for a pizza, you have sauce on there anyway. You're not tasting like this super sourness of the crust. You know, for me, I want a crust that'll actually stand up to the sauce a little bit.
Good.
We say it's all about that foundado.
Exactly. Cameron, would you mind reading what you put in the chat for people who are watching on YouTube in a few?
I think it might explain some of our discrepancy right now.
Yeah.
It says that, it's generally thought that liquid starters favor more lab activity, right? So that would give you more of that, like we were just saying. But if you have bacteria such as Lactobacillus plantarum, or like the bacteria we just described, which is in Andy's sourdough, the Fructilactobacillus Sanfranciscensis, it could do the exact opposite. So a stiff starter in that case would give you more of the flavor. So yeah, there you go. If you know, there's some benefit to knowing where your bacteria are.
Yeah.
But it explains why people sometimes, try this exact opposite thing and end up with the same result.
Yeah. Thank you. That's confounding, but also it gives us hope that maybe one day we can unlock all the mysteries, because right now we've unlocked so few.
And that's why it's so fun and interesting.
Yeah.
So fun and so interesting. All right, guys, we've reached the end. So I want to thank everybody for coming in and joining us. Cameron, I'll start with you, and James. You guys, always nice hanging out with you guys, and we appreciate all of the incredible bakers and pizza makers that have been joining us. So Jason and Andy, thanks to you guys for spending your Monday nights with us. I know that you're busy guys and you have families and businesses and wives and girlfriends and boyfriends, you name it. So we really appreciate just an hour with you guys.
Small dogs.
We love your bread, small dogs. We love your pizzas. So thank you again, Justin. I'll let you take us out.
Yeah, thanks so much, everybody. Thanks for hanging out with us. You know the dilly. Like I just said, it's all about that foundado. Whether it be people or pizza, you don't have a solid foundation. How can you build upon it? Please, please, please. This is a crazy world. These are crazy times. Stay safe, stay strong, be long with one another, the opposite of being short, because we all belong here together. Keep making, keep baking, keep burning, keep learning. And here at Slow Rise, we never, never, never get into the politics of pepperoni and pineapple. Oh, no. Forever and ever, we say pizza always, and always pizza. Pieces and slices, everybody. Thanks for hanging out with us. With love.
Thank you, everybody.